Okay, we're recording. It's November 4th. Had a few questions about some upcoming service learning events that are going to be easy to help out on. One of those is a campus cleanup that's being sort of organized by my colleague, Elise Ferrara, as far as the biology contributions. And she's working through several student organizations such as Tri Beta Honor Society, the Biology Society, perhaps PMA, and pre-vet group, as well as Green, Nichols Green. All of those are helping to clean up campus on Tuesday, November 9th from 4:30 to 5:30. So that's just 1 hour. It might go a little longer, but we're also going to be racing sunset around that time. But what's the right way to do that is to not just wait until that day and show up. You should email her so that she has a list of volunteers. So I just put her email address on the chat. That's Elise Ferrara, Dr. Elise Ferrara. You can let her know that you're volunteering for that. And you know, there might be some kind of sign up or something like that, but the sign up does not get you the credit in this course. The way you get credit in this course is after you do that, you write a little 1-page service learning event report.
Dr. LaFleur.
Yeah.
I'm actually I guess I organized it for the Green Club, but if anybody doesn't want to email Dr. Ferrara or have any questions about it, they could just email me your name because I have to submit it all to Nichols of everyone from all the different organizations.
Okay, so that's Courtney Pratini. So Courtney, in case people don't have your email, did you put it on the chat yet?
Yeah, I can put it in there.
Yeah, put it on the chat. Okay, and what I was saying was, let's say you're writing your service learning event report. And I had a few people say, I can't make it only 1 page. Yeah, yes, you can. I don't have time to tell you what sentences to cut out. Just make it 1 page for me. I mean, I realize that if you have 2 pages of information, that means you really did a good job and you have a lot to say, and I appreciate that. But the guidelines are 1 page. Just make it 1 page for me. That means if it's going over 2 pages, you have to look at that and say, I guess I can cross out this sentence and cross out this sentence, and maybe I don't have to say quite as much right here. You can do it. I know you can do it. So try to do that for me. Don't tell me I couldn't do it. I couldn't keep it to 1 page, because I know you can. I'm trying to make this easy. So that's an easy amount of points. Okay. There's another thing coming up, and that is on Sunday, November 14th. That's the rescheduled boat, weir removal boat parade. So they removed the weir this summer, and people of Thibodaux are celebrating that now. That's November 14th, which is Thibodauxville weekend. On Sunday, there's not much of Thibodauxville left, but Saturday is the main Thibodauxville celebration day, as well as a little bit of Friday. So on Sunday, they'll have that boat parade. And if you participate in your boat, that's not the same as helping to volunteer. So to volunteer, you have to write to Ryan Perk. He's the director of Bayou Lafourche, Friends of Bayou Lafourche. And it might mean that you have to show up early around noon and help him set up some tables and stuff. And it might mean you have to stay late and help people get out of their pirogues and their boats. So you let him know ahead of time, like a week ahead of time. That'll help him plan. So I added his name to the chat, I think, and it'll help him if you email him ahead of time. Okay, so those are 2 easy things. You might have some other opportunities, because we're coming up soon with some kernel days. On these kernel days, that's days that we're sort of recruiting high school students and parents. And if you help host a table like for PMA or Biology Society, and you're helping to recruit these students by telling them what we do here at Nichols, then that also works out as a service learning event. So those are all pretty easy service learning events for you to get that credit. Okay, let's see something on the chat. I think it's Courtney Pertini's email. So thanks for doing that, Courtney. All right, I see something maybe coming in from Lauren.
Dr. LaFleur, we can't see your chats that you said you put in.
Really?
Yeah, I don't know if you had it where everyone can see it. Also, there's a little pre-vet meeting over Zoom this evening at 6 p.m. People can contact me or Mason Chenier. I will drop the Zoom link in the chat right now.
It looks like I was only chatting with David LaBeouf for some reason. Sorry about that. Okay, so I'm gonna re-put mine in. That's because I started because David LaBeouf asked about that campus cleanup, whether that works, and you think you can see it now, Lauren?
I don't see anything yet. Just make sure it says to everyone. You click that little drop down menu and it says send chat to you.
Okay, I think now.
There you go.
Mine went.
Yes, we can see it.
So there's Ryan Perk's email as well as Elise Ferraris. All right, we're getting ready to start talking about physiology now. But Lauren, I kind of missed what your announcement just was because I was playing with the chat. So what was your announcement, Ms. Semino?
Just another pre-vet meeting this evening. I put the Zoom link and the meeting ID and passcode in the chat for anyone who's interested, people who are pursuing veterinary career, or maybe just want to help out animals in general, because we have lots of things coming up.
All right. Okay. Today's November 4th. We're going to continue to talk about digestion. I'm multitasking today, which means it's going to be sort of short, but there's some important facts for me to share with y'all still about the digestive system. So I'm going to do a little drawing here. To get some of the parts that we talked about the other day. There's the esophagus. Esophagus leads to, let's go this way, stomach. Esophagus to stomach. The stomach leads to the duodenum. And it's not a bad idea to practice this. You know, you could draw this in different ways, but one thing that's sort of true about the duodenum is, okay, it really comes after the stomach, and it really does kind of curve around, kind of like a C shape. And what's sort of important about that is, although the rest of the intestine, you know, you can pull it out and it's a really long tube, this little spot between the duodenum and the stomach, it helps to draw it in this C shape because this is where the pancreas lies. It is located right here close to the duodenum. So it kind of helps to draw the duodenum in this shape here, and nearby is the liver. Right, the liver is, and the pancreas, you know, they have digestive functions, but sort of like some of what we were saying the other day is they're not part of the digestive tract. They're not part of the tube. So they're considered supplementary organs They are digestive organs, but they're not part of the tract. And the liver even has this little green sac next to it. That's the gallbladder. So the liver and the gallbladder are close by. And we draw them close by like this because the pancreas, through the pancreatic duct, it sends stuff into the duodenum. And the liver and the gallbladder send stuff into the duodenum. So it's not a bad idea to try to draw these kind of close like that. All right. So if this is the duodenum, you can use your context clues to always find the duodenum because the small intestine hooks in right here to the stomach at this pyloric sphincter. Pyloric sphincter right here, right? But then the intestine, it gets pretty complicated. It's a big tube. And the middle part of the small intestine is pretty hard to delineate, right? Because what's the difference between the duodenum and the jejunum? There's hardly any that you can see with your eye. But the middle part is the jejunum. And then once again, you can kind of use your context clues to where the place that the small intestine hooks into the large intestine, to the colon. If it's hooking into the colon, then you're at the ileum. So right here, what I'm drawing is the ileum. That hooks into the colon like this. Right. I'm drawing the colon a little bigger. It's thicker, right? Where that ileum hooks into the colon is right here. You call this the ileocecal valve. And there really is sort of like this funny little dead end of the colon down here. This is called the cecum, this little dead end down here. And it's right off of the cecum that the appendix sits. Okay, a lot of people have an appendectomy. The appendix gets inflamed. So you take that out. The appendix is a little immune organ that traps bacteria. All right, so here's sort of the short version of this tube. The small intestine hooks into the colon. Now I'm going to draw the colon. In this case, I'm exaggerating it, but the colon leads to the rectum, which is sort of just for storage. The colon is really absorbing water. Once you get to the rectum, there's not a lot of water absorption. So you can tell the difference between the colon and the rectum. And then the anus is the way out. Okay, so we've gone all the way through there. All right. One thing that I wanted to mention to you, sort of in classification of digestive organs, is I wanted to talk to you about the difference between our stomach and the stomach of a ruminant. There's a lot of important animals in our lives that are ruminants, like cows, sheep, and goats. Okay, our stomach is considered to be monogastric. This is a term that has to do with the reactions that go on in the stomach. Okay, what reactions go on in our stomach is mostly hydrolysis. Hydrolysis of the peptide bond. The short way of saying that is pepsin can break apart the peptide bond of proteins. We already went over that, so this is sort of review, but that's what's going on in our monogastric stomach. That's important. But ruminants have a digastric stomach. Digastric means there's two things going on. And since these animals are pretty important to us, I want you to understand that the first thing that goes on is fermentation. Okay, in a ruminant stomach, there's fermentation, and then that's followed by hydrolysis. And some of this is in your textbook. On page 150, where this is described. Okay, so one part of this is almost confusing, but I'm going to try to say this in a non-confusing way. The digastric stomach, that's named because it's got two processes, two reactions, digastric. But in a cow, there's 4 chambers, right? These are the chambers. There's the rumen. I mentioned that a little bit the other day. You know, this first chamber of the stomach, the rumen, is why these animals are called ruminants. And the rumen allows for this activity called rumination. Okay, but the rumen is followed by the reticulum. The reticulum is followed by the omasum. And that is finally followed by the abomasum. Okay, what happens in the abomasum is pretty similar to what happens in our stomach. It's in the abomasum that hydrolysis occurs. Hydrolysis of proteins. So the abomasum of a cow is pretty similar to the monogastric stomach of a human. Okay, what about these other things? The whole reason that we have a digastric stomach in these animals is that they're eating something different from us. They're eating a lot of grass, right? And grass has a lot of cell walls. These are plant cells. They have proteins in them, but they're also surrounded by a cell wall. And much of that cell wall is made of cellulose. And our regular old stomach that is good at breaking down protein is not good at breaking down cellulose. We don't make an easy enzyme that can break down cellulose. And so what happens in the rumen is that's the beginning of a place where we culture microbes. Now, we do have microbes in our gut. So we have what is sometimes called the flora of our gut is a bunch of microbes. But these animals, these ruminants, they have a special set of microbes that are really good at breaking down cellulose first. Okay. So they ferment plant material like grass and leaves. And by fermentation, they can break down much of that cell wall that's made of cellulose. And so this is kind of like preparation for going on to the abomasum where protein can be broken down. So this fermentation step has to happen first. Now the rumen and the reticulum are slightly different. One thing is that, okay, I talked about this the other day, a cow can swallow a bunch of grass in its rumen and the bacteria can start working on it. The cow can walk to the other side of the field and not eat any grass, but he can regurgitate that grass back up into his mouth and chew it some more. Okay. You know, that's called rumination. That's regurgitation on purpose. Plus chewing, which in science we call mastication. Mastication. And, you know, mastication is not really the digestive breakdown of anything, but it's like the physical breakdown of tearing apart one big strand of grass into smaller and smaller bits. And by breaking it down at that physical layer, then the enzymes can work on it. So there's more surface area so that the microbes can begin to ferment it quicker. And then finally, the enzymes like pepsin can help break down those proteins. Okay, the reticulum is also a place where fermentation is going on, but once a cow puts grass into its reticulum, okay, it can't regurgitate it from there anymore. So the rumen is the beginning of the fermentation area where rumination can occur. So you can regurgitate, then swallow again. And then the reticulum is where more fermentation is going on. Okay, from the reticulum, that bolus, that chyme that's started to be broken down, goes to the omasum, which is kind of like a little storage area. It gets ready for things to go into the abomasum where hydrolysis continues. So this is the 4 different chambers of a ruminant stomach. And it's the place where these 2 special reactions take place: fermentation and hydrolysis, right? That's different from our stomach, where we don't use fermentation. We mostly only use hydrolysis of proteins. Okay. Now, one of the things that I tried to impress upon you is that much of what's going on in the inside of the GI tract occurs because of exocrine secretions. And today I'm going to try to separate important exocrine secretions from important endocrine secretions. That help with digestion. So Miss Rachel Henning, you with us? Can you remind us, Miss Rachel, what the difference between an exocrine secretion and an endocrine secretion would be? Looking at this picture right here.
Okay, so exocrine is basically like outside. So it's happening outside of the blood, and endo is inside the blood. So anything in the stomach is basically exo because it's not inside the blood.
Perfect. And to me, that's a hard lesson to know, because you feel like something is in your stomach. You feel like it's inside your body. But if something is secreted into the lumen of the stomach, the lumen is the inside tube, that is an exocrine secretion, right? Because it's kind of similar to saliva, which is an exocrine secretion. And saliva is kind of similar to sweat. I mean, it doesn't seem like it's similar, but like sweat on the bottom of your chin, that's to the outside surface of your body. And what's the difference between your chin skin and the skin of your cheek and your tongue here? There's hardly any difference. On the inside of your mouth, that is still skin. It's the outside of your body. And on the inside of your cheek, that skin is connected to your esophagus, right? That's still outside exocrine. Okay, so much of what we've talked about so far were exocrine secretions. The exocrine secretions go into the inside tube here. Okay, so there's an exocrine secretion here. That's amylase. I like to think of the big 3 of digestion occurs from 3 big enzymes. Okay, amylase is released by your salivary glands at the stomach. This protein here, this important enzyme is coming from the chief cells, right? And the main digesting enzyme there is pepsin. If we describe it the real way, it's like these chief cells, they secrete pepsinogen. which is the precursor. But once it gets into the acidic environment of the stomach, it is converted to pepsin. Okay, so amylase, pepsin, and then the last one that we talked about is coming from the pancreas right here, and that squirts into the duodenum right there. And what's that? That is lipase. Okay, so lipase is coming from the pancreas, that squirts into the duodenum. So we could think of digestion as more complicated, but that's a pretty good intro for this undergraduate course. Amylase, pepsin, and lipase. They are breaking down carbohydrates, proteins, and lipids. How does this get started though? The way that these digestive tubes know to secrete lipase is turned on through endocrine secretions. So what we're going to talk about now is how these get turned on through endocrine action. It's how exocrine secretions are turned on by endocrine secretions. So to do this, I'm going to draw the stomach and this pancreas a little bigger, right? You might have to turn the page and just redraw this with me, because we need a little more room here. Right? There's the stomach, which leads to the duodenum. This is the duodenum here. This is stomach here. I'm going to leave my liver and my gallbladder there. Okay. Nearby, kind of like in the C curve of this duodenum, that's also where I'm going to draw my pancreas. Now, pancreas has a big portion up here. It's almost like it's the head of the pancreas. It's really called the head, and it's kind of putting its head on the pillow of the duodenum. And the pancreas really does, it tapers off. It has this funny shape where there's the body and the tail. And, you know, in my mind, the way that I think about this glandular material, we're not going to look at it at the microscopic level yet, but there's two ways for things to go from the pancreas. One is from this pancreatic duct. A duct secretes into the lumen of the duodenum. And you could think of that as the front of these cells, because some of these cells are secreting towards the back, towards the blood, and also towards the front, which is connected to this duct here. Right? And this is where lipase is being released. Lipase is being released this way into the lumen of the duodenum. But on the back side, there's also cells that are secreting into the blood. Now, some of this is review because you already know these cells, right? One group of cells are the alpha cells of the pancreas. Another group of cells are the beta cells. These do two practically opposite things. These alpha cells secrete glucagon. These beta cells, they secrete insulin. These are two endocrine secretions that more or less work in opposition. Insulin is the endocrine hormone that tells cells in the body to take up glucose. So you're moving glucose from the blood into the cells. You could think of insulin as helping take glucose out of the blood into cells. Glucagon works in opposition to that. It starts to happen like when you're starting to be hungry, but also like during starvation. It tells you to break down glycogen into glucose and push that glucose into the blood to distribute blood, right? So these two things are working in opposition to each other, yet they come from the same organ. They come from the pancreas, and they are separated by two different cells in the aisles of Langerhans. The alpha cells secrete glucagon. The beta cells secrete insulin. All right. So let's get to the stomach. How does the stomach get turned on? Okay. The stomach has two gates. One is the gastroesophageal sphincter. Gastroesophageal, it makes sense, right? This is gastro. Stuff about the stomach has to do with gastric. The esophagus is right here. The gastroesophageal sphincter, that's what goes wrong whenever you feel heartburn, whenever you don't exactly vomit, but a little bit of stuff from your stomach goes up into your esophagus. It's because your gastroesophageal sphincter is not working the way it should. Down at the bottom, this gate here is the pyloric sphincter, kind of similar. Okay, it keeps stuff in the stomach and then releases it into the duodenum whenever enough protein has been broken down. Now, you might recognize this as one of those reactors that we mentioned the other day. This is a continuous flow batch reactor with mixing, right? That's what our stomach represents. Okay, the stomach does not just release pepsin all day, every day. The stomach responds to a bolus, right? A bolus of food. This could be like a little piece of Whopper from Burger King. What happens is the cells of the stomach distend, okay? So we have pressure sensing cells that feel stretch. So the way that this is usually described is the first signal here is stretch. That stretch turns on a different type of cell. And this cell is the G cell. The G cell releases an endocrine hormone named gastrin. It's gastrin that tells the chief cells next to it, it's time for you to secrete some pepsinogen. It's gastrin that tells the parietal cells, what are they going to secrete? They're going to secrete acid. The way they do that is to pump protons, right? You could either say that they're secreting acid, you can say that they're pumping protons. It's the same thing, right? And what that leads to is hydrochloric acid in the stomach, right? It's gastrin that also tells another group of cells. Those are the goblet cells. They're kind of shaped like a goblet. They secrete mucus. Okay. So the bolus is the first signal, distension of the stomach, but that doesn't turn on parietal cells and chief cells and goblet cells. It's this endocrine hormone that works through the blood, right? This gastrin, the way that I'm drawing this is on purpose. Gastrin is not secreted into the lumen. It's secreted in the blood, and you can imagine it going really everywhere, but these cells have receptors for gastrin, so they respond to gastrin by secreting their important exocrine secretions into the stomach. All right, so then you can kind of imagine what's going on here. We have protein being broken down into amino acids. Okay, so that bolus that looked like a little piece of a Whopper hamburger a while ago, it stops looking like a Whopper hamburger and it starts looking more homogeneous, right? As this homogeneous mixture here, you could call this now chyme, that gets squirted into the duodenum. And the duodenum was empty a while ago. It's also distended. It gets slightly distended. Okay. Now, the cells of the duodenum, they recognize, oh, there's something inside here now. And so they secrete some endocrine hormones. They secrete one that's called secretin. It was isolated and identified so early in endocrinology, it was given this very generic name. It was named secretin because it led to some secretions, right? But secretin is a protein hormone that is going to lead to some changes. It also releases another endocrine hormone called cholecystokinin. Cholecystokinin. I'm going to write it right here. Cholecystokinin. Cholecystokinin. Think there's an E in there? Cholecystokinin. I mean, I can remember some words, but this is on page 159, but cholecystokinin is hard for me to always get right. Cholecystokinin. It's cystokinin. Page 150, page 160. And the last one is GIP. Gastroinhibitory peptide. Okay, let's talk about what these do. Because the stomach has just pushed its bolus into the duodenum, what a while ago turned on the stomach now has to start to turn off the stomach. You can probably imagine how this is going to happen. This is the job of GIP. It goes through the blood, but one of its main targets is the G cells. In physiology, we draw an inhibition instead of a arrow. We draw it like this. GIP inhibits the G cells from releasing gastrin. And what that starts to do is turn off the chief cells and the parietal cells. Right? Makes sense. A while ago they were on, but now they have to start turning off because most of the stuff in the stomach is being pushed into the duodenum. That's why your stomach works for about an hour, but then it stops working. Okay. What about secretin and CCK? Secretin, like its name sounds, it's going to lead to the secretion of something. It's the thing that turns on the secretion of lipase. It's the thing that tells the pancreas it's time for you to release lipase into the duodenum. So secretin leads to the release of lipase. Secretin also has an effect on the liver and the gallbladder. Right? It's telling them it's time for you to release bile salts into the duodenum. Okay, so lipase is being sent into the duodenum, bile salts are being sent into the duodenum. Cholecystokinin is recognized for having a very similar activity, but it also leads to some bicarbonate being released. Okay, bicarbonate. So there's several things that are going on in the duodenum. One thing is this very acidic mixture that just got squirted into the duodenum, it has to be neutralized. So the way that gets neutralized is through liver and gallbladder bile salts being sent into the duodenum. And those bile salts have at least two functions. One is they work as emulsifiers. Emulsify. You got to be careful about what you say about an emulsifier. It does not digest the lipid, but it breaks large lipid droplets into a bunch of small lipid droplets to make it more easy to get exposed to lipase, right? And the bicarbonate helps to neutralize. So there's bicarbonate also in those bile salts. So neutralization is going on, emulsification is going on, and that allows for lipase that's squirted into the duodenum to start breaking down those lipids. Okay. So there's at least two steps here, right? The first step is the stomach gets turned on. The second step is the duodenum has to get turned on. But it's not really the duodenum that's being turned on. It's more like you're turning on the pancreas and the liver to send their stuff to the duodenum. And then the third behind the scenes thing that's going on is GIP is coming back around and it's turning off the stomach at that time. Okay, so that is the endocrine regulation of these steps of digestion. And that's described pretty well on page 159. There's a few other endocrine things that are going on. Okay, some of this is going to be review because I've already introduced you to at least some of these names. And these ideas of the fed state and the unfed state. Right? The fed state is what happens right after you eat. Okay, that might happen in the morning when you have breakfast, might happen right after lunch, right after dinner. One thing that happens right after the fed state is your adipose cells, adipocytes in the fatty tissue around your body, they secrete leptin. And what they indicate is that you have just taken in some lipid, right? That leptin helps to give you the feeling of satiation. It's the feeling of fullness, satiation. Now you can almost imagine the difference, right? So you sort of need some fats in order to feel full. Like an example is you can eat a whole lot of celery. You know how that is? Like I like celery, but you eat like 3 stalks of celery and that can fill your stomach, but you don't feel full unless you add some nice salad dressing on top. And what's the salad dressing have? It's got like vegetable oil sometimes or olive oil, or it's got like mayo in it, or that salad dressing. I mean, we think of it as it makes it taste better, but what it really does is because there's fats in the salad dressing, your leptin is released in your body and it kind of tells you, oh, you're full now. If you eat just celery, even though your stomach is full, you kind of don't feel full. So maybe you'll recognize that the next time you eat some veggies. It takes something extra, a little bit of dip to feel full. And I mean, the dip is adding calories. So it's some of that as well. But much of what that is the effect of leptin telling you, okay, you just took in some fats. So now you're starting to feel full. Now, another thing that's released from the pancreas, the beta cells release insulin. Okay, so you sort of know about that already. That insulin tells the tissues of the body, hey, we got a bunch of glucose in our blood now. It just came from our digestion. And now I want you tissues to take back that glucose and bring it into the cells. Okay, the cells of the brain need glucose. And so that's important. So in the fed state, these two things are going on. Leptin helps to tell you that you're full. Insulin helps to tell your tissues it's time for you to harvest some glucose. And then what? In 4 hours, you start to get to the unfed state. This is kind of like a gradual change, but we all are familiar with, you know, after you eat, you're not hungry. Then you start to be hungry. You start to kind of run out of gas. You might start feeling hangry, like on a Snickers bar commercial. So what's going on during the unfed state? That is the effect of an endocrine hormone that works in opposition to leptin. Ghrelin leads to hunger. It leads to the feeling of hunger. And it's indicating that you are starting to run out of those nutrients that you harvested about 4 hours ago. Also in that unfed state, it's the alpha cells that release glucagon. Glucagon. All right. And sometimes in a diet, this is kind of helpful. If you get to that state where you're kind of hungry, glucagon starts taking the glycogen stores in your cells, and it leads to the breakdown of that glycogen into glucose. So you start using your carbohydrates rather than taking in more carbohydrates. So glucagon leads to glycogen being broken down into glucose. And then the glucose is secreted from cells into the blood. And you know, the reason that this is happening is when you're starting to get to that unfed state, your brain is starting to run out of glucose. Okay. And your body kind of recognizes that, oh, many of our tissues are starting to run out of glucose. We need a little more glucose rather than, I mean, if the ghrelin leads to another meal, okay, that's it. But if you don't take in that meal, this is starting to happen. Glucagon is starting to harvest glucose from the cells. You're breaking down glycogen and pushing glucose back into the blood to distribute glucose. All right, I see some chats. Yeah. So someone asked about, does cholecystokinin lead to the release of bile salts? Yeah, this is what bile salts have in them. Bile salts have an emulsifying agent, and bile salts also have bicarbonate. So bile salts deliver both a neutralizing effect as well as an emulsifying effect. And cholecystokinin is the endocrine hormone that leads to that release of bile salts. All right, so that's a pretty good intro to where those exocrine secretions are working, but really what tells those exocrine secretions it's time for you to be employed in the digestive system. Okay, Miss Ivy, you have a question you want to ask?
Yeah, sorry if you answered this already. I had to walk away for a second, but you said that our digestion is better at breaking down proteins instead of like grass and stuff like cows. So what happens when our body takes in like grass, like not grass, but like vegetables kind of stuff?
Well, we can break down carbohydrates. So, you know, like rice, that's kind of like grass, right? Cereals and grains, we can break down some carbohydrates. So that's the carbohydrate component of something like rice. But our body doesn't usually break down the cellulose, like the cell wall. And so some of that cellulose, it just goes right through us, right? But that's okay because we're also eating lipids and proteins. And so we get some carbohydrate breakdown from complex sugars into simple sugars. And who does that is the amylase does that. And we're also getting proteins broken down in amino acids and triglycerides into fatty acids and glycerol. But a cow doesn't eat hardly any lipids or hardly any proteins, right? A cow eats all grass. So because it's eating so much grass, it needs some help. So it needs that fermentation step. So if we were eating nothing but grass, we would probably need some fermenters as well. And there's other animals besides ruminants that do that. Like your textbook goes into this a little bit, like rabbits. Rabbits, they don't necessarily have the same kind of stomach that a cow does, but rabbits also eat a lot of grass, and they also use fermentation in their reticulum. So that's possible. Now, the fermentation that cows do releases so much methane that a lot of climate change scientists are saying that one of our biggest sources of methane as a global warming molecule, it's coming from cows. It's coming from cows burping up from fermentation. So this is starting to be targeted as maybe there's a way that we can decrease that. And so I've seen one idea is that rather than cows just eating grass all day, if you add a little bit of seaweed to their diet, like in their feed, that seaweed, that algae, marine algae, doesn't lead to as much methane. And so there, people are looking for some ways that it's not like we're going to stop eating cows or stop having cattle, but maybe we can change the diet of cattle a little bit so that they don't release so much methane. Okay, that's it for today. Like I said, I'm multitasking. If there's something that you need to talk about, you can email me. If you need to make an appointment, it's best to email me and let me know. All right. Not seeing any chats. I hope you'll have a great weekend. Remember to wear your mask because we're not totally over COVID, but I'm glad we're kind of coming around the corner of it. All right. See you on Monday and Tuesday.